DNA replication is incredibly accurate, but mistakes still occur. Even with an extremely low error rate, the enormous size of the human genome means that errors occasionally slip through. Mutations are changes in the DNA sequence. Most have little or no effect, some are harmful, and a few may be beneficial and contribute to evolution. There are two major classes of mutations that we'll talk about in this class. The first one is point mutations. Point mutations affect a single nucleotide. There's three kinds of point mutations. Silent mutations do not change the resulting protein. Of all the amino acids in the wild or the non-mutated DNA are the same as the amino acids that the DNA strand with the error or the mutated version. Evolution doesn't care about silent mutations. So here's our wild type for a silent mutation. And here's our mutated. We were supposed to have an A. This had a mistake and called for a T, but we have the same amino acids resulting whether we have the wild type or the mutated type. So there's no damage here. It doesn't matter. Okay. In a missense mutation, one amino acid is changed. So again, here's our wild type. Here's our mutant. Okay, we're looking at this spot in the A. It was supposed to be a C. The change in amino acids can be benign if the mutated DNA calls for an amino acid that is similar in size and has similar chemical properties as the original non-mutated DNA. But it can also be very problematic. For example, the mutation that causes sickle cell disease is a missense mutation. That one change can alter the structure and function of red blood cells, which can lead to serious medical conditions in folks with the mutation. Nonsense mutations are going to call for a stop codon prematurely. The resulting polypeptide chain will not fold properly and will likely have altered structure and function. And this can be a fatal mutation. The other class of mutations that we talk about in this class are called frame shift mutations, and they result from insertion or deletions. These are also called indels. The genetic code is read in groups of 3 nucleotides. 3 nucleotides call for 1 amino acid. Therefore, adding or removing nucleotide bases shifts the entire reading frame. These mutations are often the most damaging because all of the amino acids downstream of the indel mutation will be thrown off. So here's our reading frame, but instead we're seeing this CCC is now shifted. Cells use multiple DNA repair systems. First, DNA polymerase proofreads the newly synthesized DNA during replication. We're going to make sure that as we're incorporating new nucleotides, that the nucleotide that's being added is going to be the complement of the other strand. The second DNA repair system is called the mismatch pair. These enzymes detect and correct improperly paired bases. In this case, we see that our parent strands have this molecular marker saying that these are the original. And here we can see this improper pairing. So these enzymes are going to read the original strand. They're going to find this marker. They're going to know, oh, yep, this is the original one. So what they're going to do is come along here. They're just going to read that this is incorrect. So now This T is going to be removed, replaced with a C. The third repair mechanism is called nucleotide excision repair. These enzymes are going to remove damaged DNA segments and replace them with a correctly synthesized DNA. So in this case, the hydrogen bonds are forming between adjacent nucleotides instead of the ones across from each other. So this whole segment of the DNA is going to be cut out, replaced with a functional one, and then our DNA ligase, this is our Scotch tape, will seal up these sugar phosphate backbone ends.